Anti-aging biohacking influencer Bryan Johnson has been in the news again recently, with claims he's making a Baby Bryan Petri dish version of himself. What does he mean by this? Is it legal? And what is human cloning? As a doctor of stem cell biology myself, I decided I wanted to investigate. Keep watching to explore it together. I'm Grace, and this is Grace Talks Science. Those of you not familiar with Bryan Johnson, he's an entrepreneur and the founder of the Don't Die movement, best known for treating his body like a science experiment for human longevity. His routine includes intensive health tracking, a strict regime of diet and exercise, and some more controversial therapeutic interventions. He has even taken blood plasma from his very own son. Whether you think he is crazy or a genius, you can't deny that he is a fascinating character. He's been in the news recently with a rather sensational update. I just cloned myself as a newborn. He went on to suggest that this clone could one day be used to test therapies and grow transplant organs. So is Bryan Johnson talking about growing a baby Bryan in a Petri dish? And if not, what on earth is he talking about? Ever the marketeer, he is of course exaggerating to make headlines. Good business strategy, though scientifically misleading. In fact, what he is talking about is actually far more interesting than making clones. Let me explain. The Yamanaka factors he mentions are known as the core stem cell factors. They consist of SOX2, OCT4, KLF4 and c-MYC. When these are switched on in adult somatic cells, they become reprogrammed back to a stem cell-like state. These are then called induced pluripotent stem cells, or iPSCs, and amazingly, they can be stimulated to produce many different cell types. For example, you could take a muscle cell, reactivate the Yamanaka factors, and transform it into a nerve cell. This discovery revolutionized regenerative medicine and earned Shinya Yamanaka the Nobel Prize in 2012. Researchers can use iPSCs to model diseases and study how cells develop, including testing new therapies. So when Bryan talks about having a clone to test therapies on, this is almost certainly what he means. Having a renewable supply of his own genetically matched cells, rather than a miniature human in a dish. But if he hasn't cloned himself, has anyone? Is it possible that there are human clones out there? Well, technically, yes. But before we can answer whether humans can actually be cloned, we first need to decide what a clone actually is. A clone is simply an organism or cell that is genetically almost identical to another. And despite what science fiction might have you believe, clones aren't just something produced in a laboratory. They're surprisingly common in nature. In fact, when I was about 8 years old, I confidently told everyone at school that I was a clone. This was because I'd read in a textbook that identical twins are natural clones, and having an identical twin sister myself, I'd basically proudly announced to everyone that I was grown in a lab. Needless to say, I'd slightly misunderstood the science. But I wasn't entirely wrong. Identical twins form when a fertilized egg splits into two embryos during an early stage of development. Because they both originate from the same fertilized egg, they are genetically almost identical. In that sense, me and my sister are nature's closest equivalent to clones. And we're not the only ones. Some species can reproduce without mating at all, in a process called parthenogenesis. Instead of inheriting DNA from two parents, the offspring develop from one single unfertilized egg. One of the most famous examples is the Komodo dragon, as if they weren't cool enough already. In 2006, zookeepers at Chester Zoo were astonished to find that their female Komodo dragon Flora, who had never been housed with a male, laid eggs which hatched into healthy offspring. Genetic testing confirmed that she'd reproduced entirely on her own. But how? Komodo dragons have ZZ and ZW chromosomes instead of the XX and XY that humans have. When a female has not mated, she can duplicate these chromosomes in the eggs to make WW and ZZ offspring. Whilst the WW female eggs are not viable, the ZZ male eggs can hatch healthily, happily, and as clones of their mothers. It's a remarkable evolutionary trick that allows isolated females to produce entirely new populations if necessary. I also want to say, if you're enjoying this video, please give me a like and subscribe, it really helps me out. So clearly cloning, or at least producing near genetically identical offspring, isn't as unusual as people think. Cloning in the lab, however, is a whole nother challenge. And in the 1990s, some scientists at the Roslin Institute near Edinburgh did something that no one had ever done before. They successfully cloned a mammal from an adult cell. This was, of course, Dolly the sheep, a living, breathing clone that seemed to come straight out of science fiction. Her birth sparked huge debate, both inside and outside of the scientific community. It raised a fundamental question that still follows many scientific breakthroughs today. Just because we can do something, does that mean that we should? What the researchers had achieved was an extraordinary breakthrough that transformed our view of genetics and revolutionized the field of regenerative medicine. So, how did they do it? The scientists took the nucleus from the mammary gland cell of an adult fin Dorset sheep. This nucleus contained the sheep's genetic information. They then transplanted this into an empty egg cell from a Scottish blackface sheep. This technique is called somatic cell nuclear transfer, or SCNT. A somatic cell refers to a body cell which is not a sex or reproductive cell, such as an egg or a sperm cell. Somatic cells include muscle cells, skin cells, nerve cells, and any other differentiated cells you can think of. The resulting embryo contained the nuclear DNA from the Finn Dorset sheep, so when it was transplanted into the Scottish black-faced mother, Dolly came out with a white face, so it was clear that she was a clone of the original sheep. And if you're wondering how she got her name, the donor cell of course came from a mammary gland cell, so the researchers named her after Dolly Parton. Who says scientists don't have a sense of humour? Dolly was born in 1996, and as you can see, clearly is a Finn Dorset sheep, as she has a different coloured face to her Scottish black-faced surrogate mother. She went on to live a relatively normal sheep's life. She even had lambs of her own, showing that being a clone did not prevent her from reproducing normally. Dolly was euthanized in 2003 after developing progressive lung disease. Although her death sparked discussion about whether cloning contributed to her health problems, the evidence was not conclusive. Her condition was thought to be linked to ovine pulmonary adenocarcinoma, a viral disease that causes lung tumours in sheep. And if you're interested in seeing Dolly, you can actually see her in the National Museum of Scotland. She is stuffed there today. Since Dolly, somatic cell nuclear transfer has been successfully used to clone other animals, including cattle, mice, pigs, rabbits, horses, and even rhesus macaques. So, if scientists can clone sheep and primates, why can't the same thing be done with humans? Scientifically, it's far more complicated. Ethically and legally, it's an absolute no. Although animal cloning is now possible, human reproductive cloning is prohibited or restricted across most of the world. The UN has stated that human cloning is incompatible with human dignity and the protection of human life. Additionally, there are deeper ethical questions. A cloned individual does not have the chance to consent to the procedure, and any effects on their development were therefore non-consensual. Additionally, being cloned would not just affect them, as it would also affect any offspring they have throughout their life. So, we know that human cloning is possible. Does this mean that there is a secret human cloning laboratory somewhere out there creating armies of identical people? Well, we can certainly hope not. But despite Bryan Johnson's dramatic headlines, we really don't need to worry about a wave of identical Silicon Valley billionaires taking over the world. The reality is much more interesting. He hasn't created a human clone. Instead, he's using a technology that allows scientists to rewind cells back into a stem cell state. Technology that could one day help us to develop personalized treatments and transforming medicine in the process. Thank you so much for watching. If you enjoyed this video, please like and subscribe. And please remember to mention in the comments any topics you'd like me to explore next. And as always, stay curious.